Okay, so I think probably we should get started. Hi everyone, welcome to the TMS Talk Season 3, Episode 6, and this is also the last talk of this year. First, I'd like to thank all the audience who joined us this time and have joined us before, and thanks for your attention for the past years, and for all of our speakers and the organizers to make this happen. For this specific event, can you go back one slide, Jen? Yeah, for this specific event, I'd like to thank Chen for organizing the event, and also Xiao and Bingbing and Yiran for promoting the event. And I'm the host. I'm the co-host of today's event. I'm Xiaotong from Caltech, and I'm a postdoc researcher. And before I got my PhD at Northwestern University. So for today's event, we just gonna start by a brief introduction of our society, and then I will pass the floor to Dr. Hu to introduce our speaker. The talk will last for 40 to 45 minutes, and then followed by the Q&A session. Actually, Clarice prefers to have, if you have questions during her talk, you can also raise your hand and we will promote you to the panelists, and you can interrupt and unmute yourself to ask her directly. And there will also be more time to ask your questions after the talk. Okay, for the Martlets Society, it is a non-profit organization trying to build a society for young scholars. And we have different kinds of events. We have the Martlets Talk for academia and the WBA Talk for non-academia. And we also have the TMS workshop to show you some skill set, and at the beginning of each season, we will also have a special issue. For example, this season we have Professor Teng Li to tell us about his story in his research, also in social media. And next, I will pass the floor to Dr. Hu to introduce our speaker.
Okay. Hello, everyone. My name is Chen Hu, and I'm from Lawrence Berkeley National Lab, and now I'm a postdoc. And my major is about theoretical physics. And before I got my postdoc, I had my PhD degree in McGill University in Canada. And to be honest, as a physicist, I'm very interested about this talk. And I heard a lot of this spintronics and this quantum materials before, but it's only in the physics level. But today I will I'm very looking forward to hear the talk about how to use this quantum physics to the biology systems. So this is a very interesting talk, and it's quite interdisciplinary, and I never heard this kind of talk before. And okay. Today we are very honored to have our speakers, Clarice Aiello, and she is an assistant professor in UCLA. And Professor Clarice Aiello is a quantum engineer who is interested in how we use quantum physics, and we get the information about the biology systems. And she's also an expert on the nanosensors, and which can be used to detect and harness the room temperature quantum effects in noise systems. And Clarice also received her PhD from MIT in electric engineering, and also held her postdoc appointment in bioengineering at Stanford and in chemistry at Berkeley. And she formally joined UCLA as assistant professor in 2019. And also she has her lab called the Quantum Biology Tech Lab here. And okay, so I'm very excited to hear her talk. And okay, I think as Xiaotong said, our talk you can interrupt and raise your hand and anytime during the talk, and we can give you the permission to raise hand, and we can give you the permission to speak, and you can ask your questions directly to a professor. Okay, Professor, now it's your time. Okay, I think you can start now and we can go to your page.
Thank you all. You can all hear me, right? Okay.
Yeah, very clear.
You can hear me. I can see the chat. I can see the Q&A. Awesome. Hello, everyone. Thanks for giving me a chunk of your Saturday, of your Sunday. I hope you have fun. I hope this is a fun talk and that you learn a little bit about this very emerging field that I work in. And again, as they all mentioned before, please feel free to pause me and ask questions, because I think that gets more interactive as we go. So I like to call myself a quantum engineer. This means that I build apparatuses to study and control things that are so small and so well protected from their environment that they're better described by the laws of quantum mechanics, as opposed to the laws of classical mechanics that rule everything big around us. So people get a little bit skeptic when I say that I study things that might be happening inside birds, inside butterflies. So before I explain where I'm going, I really need to tell you where I come from. So we're going to start the hour talking about quantum mechanics, and by the end of the hour, we will have talked about things of biological relevance, such as organismal migration, how our body responds to oxidative stress, how our cells interact with radiation, and I hope to have convinced you that I think we can learn with nature how to build better technologies. And that's because I'm a quantum engineer who's interested in how quantum physics informs biology at the nano scale. So usually I motivate my talk by saying that humankind is sort of obsessed with measuring things better, because this might indicate that we understand things and nature better, right? So we can think about measuring better frequencies. For example, this complex optical setup is called an atomic clock, and it sits at NIST, the National Institute of Standards and Technologies in the US, and it uses atomic transitions to define very well what a second or what a hertz is. We can think about measuring better magnetic fields so that the image of your baby is better resolved. And yes, this is the magnetic resonance image of a baby inside the mother's belly. Or we can think about how to measure better accelerations with those tiny little accelerometers so that our gaming experience is enhanced. All those tiny little accelerometers are now ubiquitous in all our handheld devices. But the question that I ask is, what happens if the quantity that you want to measure is very, very small? Or worse, what happens if the object producing the quantity that you want to measure is very, very small? In this case, I'm going to argue that we need a tiny little sensor that can measure tiny little things. If the sensor is tiny, let's make it very tiny. Let's make it quantum. Actually, there's a whole body of mathematical arguments that I won't have time to explain in their entirety that proves that if you use a quantum object as a sensor, your measurement is improved. In other words, the sensor quantumness enhances the measurement. So in my past, I have worked with a technological quantum sensor. In fact, I could put a single spin, electronic spin in diamond, in the material diamond that you have, some of you have in your earrings, in your rings. And I could, people actually can put it to work as a quantum enhanced magnetic field sensor. So I'm going to start by talking about this tiny quantum sensor that is technological. Okay, so here's the promise that a single spinning diamond can be used as a quantum enhanced magnetometer. So spin, just to refresh your memory, is a quantum property of objects that is as fundamental as charge and mass. Spin does not have a classical equivalent, and quantum objects have spin. For example, atomic nuclei have spin, even if it's a spin zero. Electrons have spin, And spin, this quantum property of spin, measures how well an object interacts with the magnetic field. And in fact, a magnetic field can change the spin energy levels. Physicists and engineers usually, and you should be looking at my image now, physicists and engineers and chemists usually represent spin with an arrow. And spin up, arrow up, or spin down, arrow down, just represent two different energy levels of this quantum property called spin, just to refresh your memory. But I'm going to show you now like how we can put a spin to use as a sensitive magnetometer, which is very weird for us to think about at first. So the spin that I work with in diamond, it arises in a crystalline defect in the diamond lattice. Diamond is composed of carbons. In the solid, there are defects, and this particular spin arises from a color center in diamond. A color center is a crystalline defect, and it's called so because it absorbs light, gets excited, and then emits light back, fluoresce. Color centers are the crystalline defects responsible for the color of those nice diamonds that you see there at the bottom of the slides. The particular color center that I worked with is called a nitrogen vacancy center. It occurs naturally, but it can also be engineered, and it arises when a vacancy, that is a missing carbon atom, sits next to a nitrogen, which is the most commonly occurring substitutional impurity in the diamond, in the diamond lattice. So when those, it's a very hyped defect, you might have heard about it, but when those two things come together, the vacancy and the nitrogen, it's like an electronic mess, right? The electrons close to the vacancy are unpaired. The nitrogen brings extra electrons. There might be nearby electron charges that get sucked into this region. So it's sort of an electronic mess. But something very interesting happens when you calculate the quantum mechanical energy levels of this defect. Almost by magic, the quantum mechanical energy levels of this defect really look like the quantum mechanical energy levels of a single electronic spin. So it's very convenient because we look at this atomically sized defect and we can consider it as one single electronic spin. The way we find those defects in the lab and play with them and work with them is quite easy and interesting, actually. It's all, even though this type of experiment can also work at lower temperatures, it has a very good advantage because we can do this at room temperature. So in the simplest of experiments, we take our diamond sample, and actually, again, now people are engineering diamonds so that those defects have better properties, but naturally occurring diamonds also have those defects. And actually the very first diamond slab that I worked with was like a discard from a jeweler. So you put the diamond slab on top of a moving stage, right? And you move it around at the same time that you shine light onto the diamond slab. In the lab, we use a laser because it's more efficient and more convenient, but actually it doesn't need to be a laser, right? We saw that with color centers, you can see sort of their colors with ambient light in that picture that I showed. And the idea is that as we are scanning the diamond sample, once the laser hits one of those defects, the defect is a color center, so that it's going to absorb light, get excited, and then emit light, fluoresce to the excite. And this emitted light we can collect so that we can take a picture of where in our sample we have those defects. And this is what it looks like at increasing zooms. So to the bottom right there, what you see is like a blob, okay? And it's quite amazing actually, because prepare yourself. This is like the fluorescent signal of one single spin. Maybe some of you have heard of nuclear magnetic resonance or electronic spin resonance. Usually it's extremely, it's impossible with our technology nowadays to get the signal from one single spin in those experiments. Here, by collecting fluorescence, we are actually looking at the signature of one effective electronic spin. Again, the defect itself is atomically sized, but the blob that you're seeing there is much, much larger because this is a diffraction limited image. But it actually gets even better because this defect, for reasons that I won't have time to explain today, but feel free to ask away later, this defect has what in the quantum sensing jargon or in the quantum community is called a quantum state dependence fluorescence intensity. This means that just by looking at how strongly that blob is emitting light, we can actually infer if the spin state is up or down. So this is a very nice way of reading out the quantum state of the particle just by counting photons and seeing if it corresponds to one intensity or the other. Yeah. But I promised you that I would give you a magnetometer out of this. So let me tell you how this works. There I depict two different electronic spin energy states. So in the y-axis, it's energy. It's like spin energy. And 0 and 1 denote here the energy levels of those two spin states, up or down, right? The energy difference between those two spin energy states are more or less in the microwave. It's about 3 gigahertz. Okay. Turns out that one of those spin states is not the one that I call 0 there, is insensitive to magnetic fields. That is, if the diamond slab is immersed into a magnetic field, it doesn't really care. Whereas the state that I call 1, on the other hand, is sensitive to magnetic fields. This means that if we put the diamond slab onto a magnetic field, the +1 energy state will be shifted a little bit by a tiny quantity called delta. And you might remember or not, if you don't remember, I tell you that because of something called a Zeeman interaction, this delta shift in energy that the +1 level in energy suffers is proportional to the magnitude of the magnetic field. And I'm going to make a point that is not usually made in the quantum sensing community. So here up, okay? So the problem of measuring a magnetic field is actually mapped onto the problem of being able to measure a detuning, a shift delta from a known energy difference, a known resonance, how's it usually called. And actually, that's how much of quantum sensing works. Usually, there are many quantities that cause shifts in known energy differences, and measuring that quantity, be it magnetic field, electric field, temperature, is actually mapped onto the signal processing problem of being able to measure a shift from a resonance. And fortunately, measuring detuning deltas from energy resonances is something that engineers and physicists have known how to do very well for many, many decades. So we can, the nitrogen vacancy center spin can measure magnetic fields because the experimentalist can measure tiny little energy shifts caused by this magnetic field. But there's a catch. The catch is that this very nice way of measuring, in this case, magnetic fields, is only valid while the spin is well described by the laws of quantum mechanics. Everything that starts quantum dies classical. And this is why we live in a sort of classical world. When quantum objects start interacting among themselves and with the environment, they sort of lose their quantum character. A spin that ceases to be well described by the laws of quantum mechanics works just like as a tiny classical magnet, right? So we can only obtain important quantum information, quantum enhanced information on the magnetic field while the spin is well described by the laws of quantum mechanics. For those of you with a quantum background, by this I mean while there is phase coherence between a superposition of the two spin states. Okay, this time of laws of quantumness sometimes is referred to as a coherence time, okay, or this quantumness time. And this time for a crappy diamond working at room temperature, naturally occurring and in bulk, is about 2 microseconds. This is, for those of you who know what this is, T2 star is about 2 microseconds in a crappy sample. People are working at, they have developed much better materials that have coherence times much longer than this, which means that we can use it as a magnetometer for much longer. But even crappy diamonds can be quantum for 2 microseconds. And even though it doesn't look like much, in the quantum sensing and quantum computing context, that's extremely long, okay? Like there are few quantum objects that can survive at room temperature as a quantum object for as long as 2 microseconds at room temperature. So in our field, 2 microseconds is extremely long. Hence, one of the why this defect is sort of hyped, because we can play with it for relatively long. And since it also works at room temperature, it's compatible, say, with biological processes and so forth, right? So what people in quantum do when they want to sort of trick nature to be able to play with this sensor for longer and longer than 2 microseconds is that we can try to kick this spin in a particular way so that it can work as a quantum object for longer. Imagine a swing, right? You have a kid in a swing. If you give kicks to this swing at particular times in particular ways, you can keep it going for long. So actually for quantum objects, it's the same. There are ways of extending, and this has been known for many decades too. There are ways of extending the quantumness time of quantum objects made of matter by the tailored application of electromagnetic fields. That is, by giving it light, kicks made of light, right? Of light, of photons, of lights, of microwaves, right? So I'm going to give you an example of how we then control this spin with photons in order to engineer its quantumness time. So I'm going to go to the most technical slide in my talk, but before that, the technical slide is not important. The important part is that you do not lose this big picture. We want to have a tiny little sensor that can be brought in very close proximity to a tiny little sample and read out magnetic fields produced by this tiny little sample, tiny magnetic fields produced by this tiny little sample. So with this said, let me go to this technical slide where I show you one example of such a control experiment where I try to keep my spin going as a quantum object so that it can yield quantum information for longer than 2 microseconds, right? So what I do, just in case there are people with a magnetic resonance background in the audience, what we do is apply a sequence of microwave pulses called rotary echo. This sequence of electromagnetic kicks, if you will, electromagnetic pulses, is depicted to the right there in nuclear magnetic resonance, in magnetic resonance jargon. It doesn't matter if you don't understand. Here's what this plot means. This plot means that what we're doing is that we are applying a continuous wave microwave onto our spin with a frequency that matches the unperturbed resonance, the unperturbed energy difference between the 0 and the 1 states. okay? We are applying this CW excitation continuously, and at particular times that the experimentalist can choose, we do a continuous phase flip, right? Electromagnetic waves have like a frequency, an intensity, and a phase. What we do is we flip the phase by 180 degrees periodically. Again, I won't have time to talk about it, but the period of phase flipping can actually be chosen to correct for different errors, can be chosen to help the qubit regain its quantumness in different ways. If we do this, if we apply this continuous wave microwave and read out the fluorescent signal that the spin gives back to us, this is what we see. The signal here is just a normalized fluorescence intensity. And if you remember, depending on the fluorescence intensity, we can read out if the spin is down or up. So for example, if we are at the top of the signal here, the spin is, say, in state down. If we are at the bottom of the signal, the spin is in state up. And every mean normalized signal, every mean intensity in between signals that the spin is in a superposition of up and down. Okay, for those of you, and this is a signal as a function of the time during which we apply these microwave pulses. For those of you with a quantum background, this is some sort of a modulated Rabi experiment. Okay, and you can already see that this signal is sort of dying down, right? When it flatlines, we can no longer obtain quantum information from this spin. And you can see that it's already like going down, but it sort of goes down a little bit more than 2 microseconds. We are indeed extending those quantumness times by applying this microwave kick of this particular kind. And we can use a technique called average Hamiltonian theory to derive to an approximation how this signal depends on this tiny detuning, this tiny energy shift delta that we talked about in the last slide, right? And this delta energy shift is actually going to give us information on the magnetic field intensity. So knowing how to interpret the signal as a function of this delta, we can Fourier transform this signal going from time to a frequency domain and obtain some sort of spectrum that you see there to the top. It doesn't, I'm not telling you how to read this. This is a bit complicated to read, but I'm telling you that looking at this spectrum there, we are reading 3 different deltas that correspond to the reading of 3 different tiny magnetic fields, the smallest of which corresponds to a magnetic field of about 1/10 of the magnetic field of the Earth. So this is like just one way that we can use this spin as a very sensitive quantum sensor. But again, it doesn't matter if you didn't get the details, the big picture is here. Tiny sensor brought very close to a tiny sample that produces tiny fields, and we can read out those fields in a very in a way that is improved over classical waves. Okay, so before we go on to the second part of my talk, I just want to mention that I have just shown you a quantum sensor that works at room temperature and in noisy environments, in the noisy environment of like close to the sample or the lattice of the diamond. And before I make an official pause and ask for questions, I would like to leave you with a question for those of you who don't have questions, right? I told you that this energy transition of this quantum sensor spin is about 3 gigahertz, right? So this is a sensor that has an energy difference of 3 gigahertz. For those of you who don't have questions, I would ask you to actually Google, right? This sensor is immersed into like a room temperature environment, right? I would like you guys to Google how much is the thermal energy in gigahertz at room temperature. Okay, I would like you just to compare the number that you get with this 3 gigahertz of energy difference of this quantum sensor. So I officially pause now. What are your questions so far?
Oh, I was asking for the previous slide, was that
Yes.
measured from a single nitrogen vacancy in a diamond or a lot of nitrogen vacancies?
No.
Sample
That was using one single atomic defect, one single nitrogen vacancy defect.
So you created that one in a large diamond sample.
So
how did you do that?
Oh no! Like our diamond sample came from nature. Nature made with a lot of different nitrogen vacancy samples, nitrogen vacancy defects. What we do, and actually you can see all of them here. Like in this middle picture here,
you
all
isolated one.
We choose an isolated one because it's simpler to work with, right? This said, first of all, there is a scheme of doing quantum sensing with a sample of nitrogen vacancy, like with an ensemble of nitrogen vacancy samples. That there are other advantages of using like many nitrogen vacancy centers as quantum sensors in an ensemble. And second point is that nowadays there's even people who know how to produce those nitrogen vacancy centers at particular places in the diamond lattice by engineering. So what they do is they bombard a diamond sample with nitrogen, okay, and then in particular places, and then they anneal the sample so that the vacancies move close to the nitrogen because it's energetically favorable. So there are ways of producing your own nitrogen vacancy defect where you want, actually, nowadays.
I see.
More questions from the audience. You're all very quiet.
Hello, can I ask maybe a simple question? So,
there are no simple questions. All questions are exciting. Tell us.
Okay, if I understand correctly, so your method is that you use a magnetic field. You can detect the magnetic field because magnetic field can induce the Zeeman effect and give you detect the level energy shift, which
Yes.
is you call the delta here, and then
Yes.
you just you can use your detector to measure this detune effect, and
Yes.
then you can get. So according to my experience, this Zeeman effect is usually very weak. So a very strong magnetic field can only induce such as a very small energy shift, maybe several milli-electron volts. So it will give you a very small detune. So is your equipment so sensitive to detect so small energy shift?
So the idea is that all of this is very fast, right? Each experiment takes like to cut a long story short, let me go back to this picture here. We are subjecting in each shot, we're subjecting the nitrogen vacancy center to like 3 microseconds of excitation, right? So in each shot, for each shot, the nitrogen vacancy center is having light for like 3 microseconds. What I didn't tell you is that we average this like almost 100K times, right? Okay. So it's the same nitrogen vacancy center, that is, you're always talking to the same spin, but we need to build up statistics. So so we do the same sequence of experiments like 100K times, 200K times. So and actually there is a way of taking that into account when you calculate the real sensitivity of your sensor, because in real life, it has to take into account how long it takes to do the whole experiment, right? Okay. So the whole experiment in this case takes like 3 microseconds times 100K times. So in real life, this is the number, like 3 microseconds times 100K. This is the number that we should use to gauge how sensitive your detector is.
Okay, so what's the usual value of your delta here? Is it maybe milli electron volts or electron volts or maybe micro electron volts?
I don't know in EV, the smallest delta here is like a megahertz, like 1 or almost 1 megahertz.
Okay.
Right, which corresponds to like a magnetic field seen by an electron of like 0.1.
Okay.
Of like 10% of the magnetic field of the Earth, which is 50 microtesla. So it's about 5 micro.
That's quite sensitive, I think.
Then again, it comes at a trade-off of time, right? It is sensitive, but you have to take into account how long it takes. It's not a lot, but this is an important parameter.
Okay, thank you. I think we have another question from audience.
Yes.
Oh, that was me. The same question.
Okay.
What's your question, Juan?
I asked you already, right, about whether it
Hello, can you hear me?
Oh, okay.
Yeah, I have so.
Oh, there's another.
Can you hear me?
Yes.
Yeah. Yeah. So I work on biophysics. So if you go back to your previous slide, the data slide you have, the micro, yeah, it so amazed me that you actually can calculate what is the exposure time of your microscopic experiment. You know, you do the laser and you use a camera to acquire the signal. So
in this experiment.
Yeah, because in our microscopy, we do a turf or, you know, those microscopy, the smallest like exposure we can get is about 3 microseconds. So it's amazed me that what you showed here.
So yes, and I hope I have time to talk about it in the second part of my talk, because yes, we need faster microscopes. So our microscope, first of all, our detection, right? We use a single photon detector, and this single photon detector has, I think, 15 nanoseconds of resolution.
Wow.
We use things to control the experiment, and this control can control things as fast as like a couple of nanoseconds, right? So yes, this is a very important point, that this is much faster than microscopes usually used, say, by the biology community. Right?
Yeah, that's amazing.
And that actually gives its power. And I'm going to argue that we need this technology to study biology as well. Thanks for this comment. It's super cool that I got to talk about it. Yes.
Yeah. Okay. Thank you.
More questions?
Do you, okay, can I ask another small question? Yeah,
always.
Did you do the control of a single, do you guarantee it's a single molecule? I mean, you must have done.
A single emitter. It's not a molecule per se, it's a single defect. Yes.
Yeah.
What you can do is a coincidence measurements, right? So what you do is you capture the light emitted by this nitrogen vaporization center and send it through a beam splitter and through detectors. And by doing correlation measurements, you can prove that no 2 photons were emitted at exactly the same time, which guarantees that you're using a single photon source, actually. So this spin can be shown, this defect that I showed to you can be shown to have been a single quantum emitter.
That's pretty cool, because I was thinking you just tell by the intensity, but you actually do a more like a delicate work. That's pretty cool. Yeah, that's
really cool.
Thank you.
So I'm going to pause here and give the answer to the questions that I asked you guys, right? You all. So sensor has an energy difference of 3 gigahertz, and it's immersed in an environment that has a thermal energy in gigahertz of about 6,000 gigahertz, right? Some of you might be confused by that. This means that, you know, like this thing that works as a quantum sensor has like a 3 gigahertz energy difference, but it's immersed in this huge, very hot bath of like 6,000 gigahertz, right? Isn't this thing going to thermalize? Actually, it is going to thermalize. And actually, when it thermalizes, it gets classical, right? So as it thermalizes, it's no longer well described by the laws of quantum mechanics. That is, this sensor, which is a very tiny, it has a very tiny energy difference in a very hot environment, only works well as a quantum sensor before the thermalization, right? Which is sort of on the same order of magnitude of those 2 microseconds that I quoted before, right? So yes, there's this huge mismatch between thermal energies and the sensor energy, but again, this sensor only works well as a quantum object before thermalization time. So there is no paradox in the sensor being immersed in a huge, like in gigahertz, a huge heat bath at room temperature per se. So there is no paradox, and because the same question might come up in the second part of my talk. Again, I have just shown you a quantum sensor that works at room temperature and in noisy environments. And up until a couple of years, those were the types of sensors that I was used to dealing with, right? All very much technological. But at some point, I sort of realized that many times nature came up with sensors that overperformed humankind-made sensors in crazy ways, right? So I'm going to argue that a magnetic sensor made by nature, right, with the stamp made by nature, organic, non-GMO, is a quantum sensor that works pretty much in the same way as the nitrogen vacancy sensor. Okay, so this is the main part of my talk and what I'm really excited to talk about. So, but before we can talk about biology, we really need to talk about biology at the nanoscale, namely the chemistry. And it's actually known from basic chemistry, like chemistry of reactions in a test tube, that magnetic fields can alter the final products of a class of chemical reactions that depend on spin. So it's known that there are some chemical reactions that start occurring, and at some point there's an injunction and there's a certain spin, and depending on the state of the spin, if the spin is up, the chemical reaction continues through one branch. If the spin is down, the chemical reaction continues through another branch. And magnetic fields can actually change the probability of us finding this electron spin in the up or down state. Hence, chemical reactions can shift if the reaction continues through this pathway or this pathway. Importantly, the final products of those two pathways are macroscopically different. Okay, so a finicky quantum effect at the very middle of a chemical reaction can have significant macroscopic consequences on the outcome of the chemical reaction. So this has been demonstrated at room temperature, in solution, in the gas phase, in the solid phase, and for magnetic field strengths down to the strength of the magnetic field of the Earth, again, which is tiny, which is 50 microtesla. Okay, 50 microtesla is smaller in intensity than the magnetic field that you feel when you bring your cell phone close to your face. Okay? How does biology come into this? Okay. Biology comes into this with birds. Okay, I promise you that we're going to go away from birds very, very soon, but it all started with birds. And birds, migratory birds, when they migrate north-south, they are known with no doubt to depend on the magnetic field of the Earth to do so, even if as a partial cue. And it has never been really understood for a long time how they do this. Again, 50 microtesla The magnetic field of the Earth is tiny. It's ridiculously tiny. So how might they be doing this? Many hypotheses had been tested by putting magnets trying to find magnetic particles in birds, trying to see if magnets, like strong magnets, could mess up with the magnet's sense of orientation. And actually, there was no good hypothesis standing on how birds do this, right? How birds use the magnetic field as a help during navigation. And then in the 80s, some very brave, almost crazy theoretical biophysicists made a hypothesis, right? At that point, it was a crazy hypothesis. Here's what they said. They said, well, if these same type of spin-sensitive chemical reactions were to be happening inside the bird, right? Birds and organisms in general could be sensitive to magnetic fields to the extent that they could be sensitive to different physiological concentrations of products coming from those two different branches of chemical reactions. For example, it was proposed that maybe there's some sort of pigment in the bird's eye, right? That depending on the way that the bird is looking at, might suffer a modulation in photosensitivity, so that maybe birds could, as they change directions, maybe they could effectively see, right, magnetic field lines. So whereas this hypothesis has not been proven yet, this photo modulation hypothesis, people started looking for proteins, and at that point, because it was birds, animal proteins that could sustain those spin-sensitive chemical reactions. And at that point, the only animal protein that was known, today it's way broader, but at that point, the only animal protein that was known to be able to sustain those spin-dependent chemical reactions was a protein, a flavoprotein called cryptochrome. Flavoproteins have a flavin, which is a pigment that is something that absorbs light, gets excited, and then fluoresces. Cryptochromes absorb blue light. So birds have cryptochromes in their eyes, migrating butterflies have cryptochrome in their antennas. We have cryptochromes in our eyes, and wait for that, we have cryptochrome in all our cells. I've checked with many biologists. This is like a good statement. We have, I do not know about reproductive cells, but apparently we have cryptochrome in all our cells because cryptochrome is responsible for circadian rhythm regulation. So and this is not weird science. If you travel across time zones and you're jet lagged, sometimes people say, look at blue light to sort of regulate, to help with the jet lag. That's partially what you're doing is sort of giving blue light for your cryptochromes to function. Don't ask me exactly how it works as a circadian rhythm regulator, but this is really known like in the circadian rhythm community, right? And actually, cryptochrome is expression of cryptochrome is very widespread across the tree of life. All those organisms there produce cryptochromes, and very interestingly, at the big scale, the organisms boxed in red are organisms for which there have been magnetic sensing studies that somehow, I'm not saying unambiguously, but somehow implicated a cryptochrome on magnetic field sensing. And given all the science that has been done for like almost 40 years now, the evidence for cryptochrome-based magnetosensing, again, cryptochrome is just the most studied pigment. We're still at the birds space. It's going to get much more general in about a couple of slides. But evidence for cryptochrome-based magnetic field sensing in organisms is really widespread, but unfortunately at very disconnected length scales. Okay, so to the right, there's like evidence at the macroscopic scale. Okay, and to the left there's evidence at the test tube scale, Okay, with the evidence in the left, I'm going to argue that cryptochrome is actually working as a quantum sensor in pretty much the same way that the spinning diamond is. For example, in the top left plot, what researchers did was record the fluorescence emitted by cryptochromes in solution, okay, fluorescence, as a function of the time during which they applied blue light onto those cryptochromes. Again, cryptochrome absorbs blue light, gets excited, and then fluoresces. The first thing that you see is that this fluorescence is going down with time. This means that we're photo bleaching the pigment. That's okay. The laser is just like photo bleaching everything. But very interestingly, as the researchers pulsed a tiny magnetic field up and down during this experiment, they saw that the fluorescence intensity of cryptochrome was also modulated up and down, as you can see in the inset. Again, if you remember, with the nitrogen vacancy center, just by looking at how strongly it was fluorescing, we could infer if the spin was up or down. Here, it's sort of the same thing. Just by looking at how strongly this cryptochrome is fluorescing, we can sort of infer if the chemical reaction continued through one branch or the other spin branch. Okay, this is not an artifact. This has been tested with different magnetic fields. And also there are studies which are not conclusive, but which seem to indicate that at room temperature, cryptochrome seems to be coherent, seems to be quantum for about 1 microsecond. Again, which is extremely long for something that is nature made, and on the same order of magnitude of the 2 microseconds that I cited for the spin in diamond. However, the next step on the length scale is like experiments with like whole birds, right? Those are extremely beautiful, extremely difficult experiments, but done like during 2 months of migration season with like n equals to 23 birds, right? So people put birds in cages and they want to see where they want to go out to migrate. And by putting like coils around and by messing up with the magnetic field that the birds see, the birds want to go out through another way, giving results that are consistent with the spin theory. Flies, don't ask me how they do this, but flies can be trained to find food via the presence of a magnetic field. This is magic to me, but they can train them, right? The flies are trained to find food when there's a magnetic field. Then the researchers knock out the cryptochrome gene from the flies. And then the flies are no longer able to find food via when a magnetic field is present. Then in a further experiments, researchers put back human cryptochrome in those genetically modified flies, and the flies are back to find food when the presence of a magnetic field. Okay. So all of this is sort of, most of this evidence is consistent with the spin model, but it's super hard to say, well, the bird is doing its migratory things because there's a quantum process that actually makes a difference that is occurring in it, right? So like it's like those are very disconnected length scales. And here's what we're starting to do. We want to bridge those length scales in our lab at UCLA. So what we want is to start doing quantum-inspired experiments inside, say, one cell, a couple of cells, maybe at some point in tissues, do a ground-up approach, right? To try to see how spin physics is actually influencing physiological phenomena, like at much smaller length scales. So what we have is basically glorified microscopes with coils. Okay, those microscopes will work much faster. We started at UCLA in November of 2019. Like 2 months later, we were all sent back home. That's not ideal for a group that builds instrumentation. So we're very much delayed, but we're building 4 different types of microscopes: one lattice light sheet, one total internal reflection microscope, one electrophysiology setup, and one STM, scanning tunneling microscope, with coils. Okay, so what we want to do is to use the coils to alter the spin state, say, of cryptochrome inside the cell, right? Try to read out if we're preparing the spin state in a way that we want, using the fluorescence of the spin, just like we did with the diamond. This is this is called optically detected magnetic resonance. And at the same time, albeit at a different time scale, at much longer time scales, correlate the spin state with what happens using, say, standard fluorescence dyes. So spin dynamics fluorescence is very weak and it's very fast. It occurs from nanosecond to microsecond, and usual microscopy markers work at like millisecond to second length scale. So we're going to collect those time scales in the same apparatuses and try to see whether spin manipulation yields something downstream. For example, how much the cells produce reactive oxygen species, how much the cell is doing something or other, right? So what we want is to use a quantum approach to those spins in proteins by treating them as bona fide quantum sensors, because that's what they seem to be. They seem to be bona fide quantum sensors. And but my talk would be very limited in interest, right? And now we're going away from birds. If this were only a phenomenon that was restricted to birds, birds are very, I thank birds because birds started this whole shebang, right? But it seems now that there are many biological metabolic processes that, again, at the macro scale, are consistent with being dependent of spins, okay? And this is much more general than thought before. And those phenomena, some might be related to cryptochrome, but some might be related to other spin radicals or other pigments that are not in cryptochrome. Okay, so this is a field that is just starting. For example, physicists, what they did was they shielded against external magnetic fields a biological machine called a metabolic analyzer. In a metabolic analyzer, you put up like a cell of thousands of a plate of thousands of cells, and you read out some products that they're producing, right? So physicists shielded one of those metabolic analyzers against external magnetic fields, put a plate of cells inside, and applied inside weak magnetic fields, okay? And just by changing the direction and I think the frequency of the magnetic fields, they could see macroscopic reading changes on like glycolysis rates, respiration rates in cells, cellular acidification rates or acidification levels, right? In a way that is consistent with the production of in a way that is consistent with things depending on spin processes. This is just one example. Stem cell in planaria. Planarias are flatworms that are very interesting for medicine because they regenerate. You cut them, they grow themselves again, right? So it's very interesting for regeneration medicine. Planarian growth seems to be seems to depend on magnetic fields in a way that is consistent with it being dependent on a spin dependent chemical reaction. Okay, I think now is a good time for me to talk about weak magnetic fields. I won't have time to explain, but all this spin physics that I've talked about is a weak field physics. This is fundamentally different with the spin physics that happens, say, inside a magnetic resonance machine. Okay, so for reasons that we can go into details, like in the question and answer, at say 10 times the magnetic field of the Earth, more or less, usually depending on the protein, but like at 10 times the magnetic field of the Earth, the importance of this spin physics effect has already drastically decreased. So all these effects that I'm talking about for birds, for this metabolic things, are weak magnetic fields phenomena, which is sort of crazy and scary because weak magnetic fields are sort of produced by a lot of things around us, right? So for this spin physics phenomena, it doesn't help to go to like very large fields at all. Another thing that depends on weak magnetic fields is DNA repair, the cellular autofluorescence. It's a recent paper from Japan. The cellular autofluorescence probably coming from flavoproteins in mitochondria is also magnetic field dependent in a way that is consistent with the spin model. Right, so it's been, I have a friend. I cannot show the picture because it hasn't been published yet. So I have a friend who's like a hardcore precision measurement physicist. So what he does for a living is he builds hypomagnetic chambers that shield very, very well magnetic fields, and then he puts experiments on precision measurements using ultra cold atoms. It's not important, but he makes hypomagnetic chambers that have magnetic field levels of noise of about 1 nanotesla. Again, magnetic field of the Earth are already tiny, 50 microtesla. This guy can make a noise floor in his boxes of 1 nanotesla. He was hearing all those things about magnetic fields in biology. So what he did, again, he's a physicist, but this experiment has been reproduced now by another physics group in Mainz and a bio group at Tufts. What they did was grow for 2 days tadpoles inside, like the little frog embryos or froglings or whatever, tadpoles inside those hypomagnetic chambers for 2 days, because it was as long as they could go without asking for a bio license or something, under 2 conditions. One, only with the 1 nanotesla floor of magnetic field, and another with like within the hypomagnetic chamber with a magnetic field inside that mimicked this like 50 microtesla-ish magnetic field of the Earth. The tadpoles grown under the mimicked magnetic field of the Earth, macroscopically they were okayish, and the tadpoles grown under the noise 1 nanotesla level, macroscopically, 30% of them were all deformed. Okay, they were, some of them were like 30% of them, some of those 30% were viable, but 30% were really like deformed in some way. This does not prove that this is a spin-dependent effect, but given the magnitude of the fields involved and the fact that you're not adding, you're just subtracting a tiny field, right? Everyone's best guess at this moment is that it really is spin-dependent. And again, this brings all sorts of questions, right? What's the magnetic field on Mars? Can we grow lettuce on Mars? Can we reproduce on Mars? So it goes way beyond birds. It possibly has a lot of implications for biology in general. So here's what we're trying to do then in our lab. We're trying to for the first time, establish or refute this correlation between spin physics and relevant biosensing biological phenomena, right? We want for the first time to measure those 2 things at the same time at smaller scales to try to see if quantum physics really can or cannot have macroscopic effects on how biology works. And we think that this knowledge can be used to manipulate physiology, for example, for therapeutic advantage, right? Imagine that, and those things actually we can model, okay? We can model at different levels of refinement, but we can model those things using standard quantum mechanical techniques. For those of you in the audience, we can solve the Schrödinger equation in the presence of noise, called the Liouville equations, to sort of model to some extent this spin physics and try to compare those predictions with what happens experimentally. I'm dying to take some experimental curves on things that we have modeled, for example. And we think that with time, not in 5 years, not in 10 years, but say 20, 50 years, we will have learned maybe, you know, like, well, if you apply this magnetic field of this frequency, direction, and intensity, this is what is going to happen physiologically, right? So imagine that you could direct physiologically those chemical reactions that depend on spin to, say, alter the outcome of disease and so forth, right? I think there's a lot to be learned there by doing those quantum-inspired experiments. And the other thing, I also think that there's a lot of high-tech, a lot of ways in which high-tech can benefit from learning with nature. For example, if nature is using quantum mechanics to work, it knows darn well how to deal with noise, right? So I'm not saying, well, let's replace Google's beautiful quantum computer with a molecule, but I'm saying, well, are there strategies used by nature that can be adapted to be used by existing quantum computer architectures to make quantum computers live longer as quantum objects, right? So that's why I think quantum biology is a very cool thing. And there are other people who are already trying to apply high-tech ways of driving and controlling those endogenous quantum mechanical degrees of freedom in biology, right? So we're not the first one, but all those experiments that I cite there, they're very recent. So that's something that's really starting and emerging. So I'm not endorsing nature, but I'm endorsing discover of nature. I really think that interdisciplinarity is what's going to help us solve big questions in the very near future. It's sort of tricky, right? Because I as a physicist, I am always learning all the time about the biology, about the chemists. My chemist colleagues and team members, they're all the time learning about the physics, about the biology. So it's really cool that you can keep learning. And I think that we are going to gain a lot as scientists as barriers between disciplines start to coalesce. So before I end, I would just like to thank my group members who are amazing scientists, overwhelmingly brave because they agreed to start a new group during a pandemic in a subject that is often not well understood so far. If any of you or your colleagues are interested in talking about possible collaborations, let's talk. And I think that's all I had to tell you today. So may the quantum be with you. What are your questions?
So can I ask a question first? So previously for the diamond center in the nitrogen vacancy in the diamond, there's a problem with thermalization. So is there any assumptions or like explanations why that could be not a problem for the cryptochrome?
So for the diamond, it takes a slightly longer time to thermalize because there are not a lot of energy levels in the environment nearby that match like the energy that can exchange energy. And with the cryptochrome, it's sort of the same thing. In all those systems, the main source of noise is spin noise, usually spin noise. It's like the spins in the sensors talking to nearby, usually nuclear spins, okay? It is thought, again, I don't think the evidence is complete for cryptochrome, but it's thought that the molecular arrangement, like the part of the cryptochrome where the spins of relevance interact. Actually, I made the thing with one spin for cryptochrome. It's actually two spins. If they are in a triplet state, one pointing up, one pointing down, the chemical reaction goes through one path. If they're both pointing in the same direction in a triplet state, the chemical reaction continues through another pathway. So I didn't tell the whole truth. It's two spins for cryptochrome. It's thought that the local environment where the two spins interact, it seems to be in like a pocket, a protein pocket, I'm told. And this seems to shield other spins from being too close for the electrons to mess up with them and make them lose coherence quicker. This seems to be the explanation for cryptochrome, but again, it's not absolutely understood yet.
I see.
And again, it's not only cryptochrome. Cryptochrome is just the main studied protein. There's evidence that other proteins also have similar spin physics. So we're going to start our studies with cryptochrome just because it has been more well studied historically because of the birds. So thank you, birds. But with time, we really want to expand and look at other proteins with interesting spin physics.
Okay.
Yes,
Yeah,
tell me. Your question was not finished. Yes.
Oh, I have another question. So, so for a lot of the biosensing and labeling process, we really need nanoparticles modified to target certain groups in the existing like nanoparticle targeted delivery-based systems. So, are you also thinking about making, say, either nanoparticles of the cryptochrome type of the or nitrogen diamond nanodiamond with nitrogen centers to do that?
So actually, nanodiamonds that contain only one, like statistically speaking, only one nitrogen vacancy center already exist, and they are as small as like single-digit nanometer. And what people are actually starting to do with those, especially in biological contexts, people shoot them into cells, and they're starting to try to do temperature measurements into cells, and maybe at some point magnetic field measurements into cells. But yes, it's already commercially available, those nanodiamonds with statistically speaking, one single NV center per nanodiamond, right? For the cryptochrome, no. What we hope to do in the beginning is to, with the help of some friends, because we cannot do this, genetically modify, say, bacteria to have like a sac, a little thing with a lot of cryptochromes inside first, right? But again, with time, we would like to use cryptochrome as it appears inside the cell. But again, this will take time, right? We're just starting.
Okay. Yeah, that's all my questions.
Okay, our next question from Chen. So can you unmute and ask?
Hello. Yeah, hello. I have 2 questions. So first, you mentioned that you want to express the protein in the bacteria and use that.
Overexpressed. Actually, some bacteria produce this, I think. So we want to overexpress it to have like a little bag only of cryptochromes.
I don't think it's expressed in bacteria, as long as I know, because it's found in plant and animal. I just wiki it. Yeah.
So what I know, again, I might well be wrong, is that like some purified cryptochrome. So you can many times get purified proteins by overexpressing them in bacteria and then just taking them out in some way. But I think, yes.
But that is after genetic modified, I guess.
Yes.
So,
you're completely right that it doesn't make sense to physiologically look at bacteria, but a cryptochrome in vivo has never been studied in any way. So one simple thing that we could do is try to see whether cryptochrome in a physiologically relevant environment could still work as a quantum sensor for as long as 1 microsecond, or try to see for as long for how long its quantumness can survive in a biological environment, for example. But you're right, the interest is not using those genetically modified things. The interest is try to do things like to study cryptochrome in cells where it occurs.
Yeah, but I see what you mean. So I think, so I'm a biologist, so I think I think first, I don't know if you know Chen group in Peking University.
No, what do they do?
He actually published a paper in 2015 about this this protein, about chromatin in Nature Communication, I guess, if you look at it. And I think they have some project about express the what is the protein name?
Cryptochrome.
Yeah, in E. coli. I think they had those projects, but I don't know if it's. I don't think they do. Yeah, I don't think they do. I can send you the name. So it's a
yes,
group.
Please. Can you please send me? I would be super happy to.
But I don't think they have the fancy measurement that you established. So the thing, my comment on the overexpressed in E. coli and then use your microscopy pipeline is the overexpressed protein. It's highly abundant in the E. coli, and you couldn't imagine the single molecule, single protein, which is crucial for you.
It depends. It depends. You are right that the ultimate goal is to do single molecule experiments, right? But in the same way that some people do magnetic field sensing with an ensemble of NV centers, we can also do the same thing with like an ensemble of cryptochromes, as is actually done with like cryptochroming solution, right? So you're absolutely right. The end goal is to do things with like one single cryptochrome. But in the beginning, I think that it's important to try to check in a very rough way whether cryptochrome can survive as a quantum object and for how long it can survive as a quantum object inside a living organism, right? There is absolutely no study about that. And if we find, like, not only in one experiment, if we have enough evidence to show that no, cryptochrome cannot work as a quantum sensor inside physiological conditions, well, then we will have data to maybe question the model, right?
Yeah. But so we did our group did some fluorescent microscopy with E. coli, and I would say it's autofluorescent by itself. So I don't know like what is the intensity of your.
Yes. So the key here is the time scales involved.
That's true.
The photophysics of spins is very, very fast. So that's the beauty of this. We have two different. It's the spin physics fluorescence is very weak. So we need actually what we're going to have in our microscopes like a dual detection. One that reads very faint signals with single photon detectors and fast with nanosecond resolution, and another one that reads like the usual.
That's pretty cool. Yeah. And I think I was thinking like how could you? I was thinking when you talk like how could you get the single protein? You actually there is a method that I came up that you can actually scissor the protein with beads on the slide, and use the turf microscopy, and you can then measure. You can then do the detection of that single protein.
Can you send that? Yes.
Yeah, you use a flow cytometry. I don't know if you're familiar with that technique in biology. So
how's it called?
Flow cytometry. Flow is a water flow, and
so they're going like this.
It's a cover slip. You have your cover slip, and you have a beads. The beads will catch certain like antibodies, and you modified your protein a little bit. You modify the gene and let it linked with the antibody, so that when it's expressed, the antibody will catch the beads. Cool. And your like chrome protein will be like it will be one single one single that protein on that bit, isn't it?
Cool.
Because you use flow cytometry to wash out all the other proteins.
But that's like outside the cell, right?
Yeah, that is in vitro, but yeah, but that
But
is
that's
the way
cool.
that you can get single protein.
Would you please also
yeah,
send me a reference for that?
Okay, I don't know if there any reference of flow cytometry because it's a really very old technique in biology. I can send you the name.
I've known flow cytometry to do like when you separate populations of cells, but not like to do protein stuff.
Is this a name? I think I will check. I will double check it. Maybe
Awesome.
It's just to wash away other
cool.
proteins. Yeah, I can
Awesome.
find some reference. Okay.
Thank you.
Okay, no problem. Yeah, it's very interesting talk. I really learned a lot.
And I need the help of biologists. I'm not a biologist. I need to learn more. So if you're interested, let's talk.
It's really interesting. Yeah, I can definitely send you the thing I was thinking about.
Cool. I'm going to put my email in the chat. It's easy. It's just cla@ucla.edu. Okay. And anyone here in the seminar, feel free to contact me to talk about it more.
Yeah. Anyone else in the audience that have a question? Okay, I have a question. So,
yes.
So in your second part about the like magnetic field, that's like very interesting. You talk about the assumption of like how birds sense the magnetic field of the Earth, which is very weak. And the assumption is about like that magnetic field can like influence a chemical reaction. But I was wondering like how could the chemical reaction be so sensitive to the magnetic field, since there are also other signals like from like the, as you said, like the cell phones, from like the other like background noise. How could that detect the magnetic field of the Earth like from all the background noise?
Several things to your question. First of all, I just want to make sure that in birds, for example, that nobody knows, there is no good hypothesis of like what this chemical reaction is. Nobody knows, right? Like in cryptochrome, like in vitro it all works, but nobody has any idea what's like the second step that might be going on inside birds. This is why we want to do like from bottom up, because a bird is too complicated. So the second part of the answer is that there are people looking at the influence of like cell phone towers and stuff onto birds, right? What I can say is that the model predicts different magnetosensitivities of the protein depending on the frequency and on the direction and on the intensity. Maybe the frequencies of like the cell phone towers are frequencies that do not produce any interference. So I don't know. People are looking into this. The third thing I want to mention is that I have simulations. I mean, and other people have similar simulations too, on how the magnetosensitivity of cryptochrome varies as a function of magnetic field strength. Right. So it's like magnetosensitivity, how strongly the protein reacts to changes around that central magnetic field as a function of magnetic field strength. At least 3 people in the world have made these calculations, and we all sort of agree. This curve is not monotonic, it goes up and down. For cryptochrome, about 10 times the magnetic field of the Earth, this curve has been divided by, the magnetosensitivity has been divided by 5-fold, okay? And it's very cool because more or less the peak of the magnetosensitivity, the peak is very close to the magnetic field of the Earth, which could be a coincidence, or it could be that there has been some evolutionary pressure. We cannot tell those things apart, but we think that people who can do directed evolution of proteins could maybe help us elucidate more about this. So
cool. Yeah, I
it's
think
cool.
you are working on this like so interdisciplinary field, and you need like knowledge from all the fields like to solve the problem. So that's really
Right?
cool. Yeah.
The other day I was talking to a geologist because sometimes the magnetic field of the Earth changes relatively fast, and we were trying to find out whether there's data that birds' migratory paths changed when the magnetic field of the Earth changed, and we couldn't find. But there's some like weird stuff that.
Cool. Are there any other questions from the
Can I ask maybe final question? Yeah. Okay. So in your slides, you draw a picture that maybe I don't know if it's a very beginning illustration with your detailed plan. Is that you want to replace this protein, this magnetic sensitive protein, to your previous diamond. Defect diamond, and to use your optical method to measure this. So because I'm thinking that for your this single atomic defect, this diamond, this one, this is you have a very pure single atomic level, energy level. But for proton, it's much more complicated. You have a lot of energy levels. So yes. How you can distinguish which energy you should pump your systems? Because you need to choose the energy to pump, right?
So the first thing that I want to say is that in the same way that we could apply light to extend the quantumness of the nitrogen vacancy center, we want to also, again, to apply light to extend the quantumness of cryptochrome. So the energy levels that are of interest are some there are different things that are occurring inside the protein, right? There are many like vibrational modes that are very fast, but here we're talking about the energy levels of a couple of spins that live inside the protein. So it's sort of very different from like vibrational energy levels. So the simplest model that people use to model this spin phenomenon like with 3 spins, but some theory people use up to like 21 spins. So what we do is we forget about everything else, which is an approximation. We forget about the structure of the protein, and we just code the spins that interact and how far apart they are on average and so forth.
Okay, so there are several magnetic atoms in the protein, which is sensitive to the magnetic field. Right?
No. There are 2 electron spins, and that is the beauty of it. So there are 2 electron spins. It's not one. Again, I told you the incomplete truth that define the fate of a chemical reaction. Those 2 electrons, they're both electron radicals that meet for a short time.
Oh, okay.
Yes,
I see.
Yes, they're called radical pairs, right?
Okay.
And most chemical reactions that depend on spin depend on the spin of radical pairs, that is of spin radicals that come together for a short time.
Oh, so that means that there's no real magnetic atom inside this protein.
No. Sorry if I misled you. No, it's not a magnetic atom or anything. It's like just those 2 electrons interacting for this brief time and forming this radical.
Something like open shell of the molecular. So it's not a closed shell. There's unpaired in electron like this.
Yes, and those 2 electrons, they don't even have a bond.
Okay,
this comes from one end, one part of the molecule. This comes from another end, and they're temporarily very, very nearby.
Okay, I see. That's interesting because maybe I had a very long time misleading. I saw a misunderstanding. I thought this protein has a magnetic atom inside it.
No. Maybe I'll start stressing this more, but it's not. There is no iron. No, like that's iron free. It's an absolutely iron free. It depends only on the temporary, like nanosecond to microsecond.
It's just an organic atom such as carbon the hydrogen or this kind of atom?
Sure, if it helps. One electron comes from the flavin, which is the pigment, and the other electron comes from a, oh my God, I forgot. The tyrosine? No. The other electron comes from like a residue nearby whose name I forgot. Flavin, and the other one is tryptophan. Tryptophan, yes.
Okay.
One electron comes from a flavin, the other one comes from a nearby tryptophan.
Okay, that's interesting. I think, yeah, there's no magnetic atom, but it can be so sensitive to the magnetic field.
For a short time, but yes.
Okay, thank you. I think my question is over, and thank you very much for your answers. Yeah, it helped me to understand this correctly.
Oh, very nice questions. Thank you all for the engagement.
Okay. If there's no comments and questions from our audience, maybe today our talk is ending. So Xiaotong, do you have other comments?
No, just I want to thank Clarice for the wonderful talk, and thanks everyone for being here. And stay tuned for our talks next year. There will be more to come. Thank you. Have a good night, or have a good day.
Enjoy your weekend. And Jen, go to sleep.
Okay, yeah, I will. Thank you.
Thank you for having me. Bye, Martlets.
Bye.